back of the vehicle as close as we can where it still maintains the auto function. And then you can actually see something in the front.
I think maybe I'll just change to north for the transect, and then I'll...
What's that?
I think we'll go north for the transect. Changing it
North.
a bit.
Yeah. Okay.
Yeah, there's no magic to the points. They were just kind of predetermined directions. So
Okay.
we can change it up depending on the terrain.
Sure.
Yeah, get a little better video there as we go up the hill.
Yeah. Let me know when you're set to start.
Yeah, I'm still playing with the
Yeah, no worries.
alti. Oh, I know that's too low. I'm gonna be contact. Okay, that's looking something.
Yeah.
I keep trying to tilt my camera and hitting the vert. That's why it comes up. Then it comes back down. Okay, I'm ready to hit the button if you, boat's
Sure.
moving, right?
Yep, boat's moving.
He's not moving north though, is he?
I'm going to change it right when we start.
Roger.
So I'll call that the start there. This is T6B
Copy.
bridge nav.
200 meters dialed in.
We'll cancel that move and let's step 100 meters bearing north, speed 0.3. Thank you. 1827 T6B. That's what I said, right? 3H1.
And this will be transect 6, is that correct?
Yes, that's what I have. Is that correct, Steve?
That's what I have also.
Okay.
I like hearing that.
Let me double check.
Last one we did, I think, was 5 there.
What's the last transect target you have?
I have to confirm with Ben because he didn't have an end target, but the last start was 5. So yeah, 5. So I just have to figure out which one of these is the end. So this is 6.
That's correct. Yep. Confirmed.
Roger that.
Okay, so Jess, we just started the transect. Looks, I don't know if we're moving forward, are we? Hey, Jess. Just started the transect, so we're headed north right now.
Sure thing.
Yeah, 0.3. And I called in a 100-meter move. We'll have to do another 100 meters. So it'll be 200 meters total. And I think the terrain should look like this the whole time.
Roger.
Okay.
Sounds good. Thanks, Ernie.
Yep.
So it's the way of transects. Once we get on transect, we go to mud and urchins.
Yeah. Perfect timing.
It's easier to count back at the lab, though.
We're doing this all to make your life easier, Tom.
Yeah, it will be fun to have some corals and sponges, even fish. There's really nothing right in this moment.
I think, but
Squat
we just
lobster.
were in a great, yeah. But we were in a great
Yeah.
habitat just now to sample, so it was perfect.
Yep, I mean, that's the whole point of what we do this for, is we just go out and randomly select areas and we just do the habitat. So there's a wreck sole that's under the ground or halfway buried. Looks
Yeah.
like a big fin eel pout coming up.
How could you tell so quickly that that was a buried wreck sole? Was it just by the little bit of the dorsal fin that you could see, or is it just the habitat that we're in?
The pigment pattern that was on its side that I could see.
Wow, there was just the smallest of small patch that I could see.
Yep,
That's pretty
that's all you can
impressive.
see. There's the siphonophore coming up that we call a pineapple. This orange,
like
yellow
kiwi
one.
bottom.
Like that. And its tentacles drag on the sea floor and it eats things off the sea floor.
So is this just an undescribed species that you call it the pineapple siphonophore, or does it really have a?
Oh, it's described, but we
Okay,
do.
good.
It's way easier to say pineapple than do a genus and species.
I agree. Whatever, and as long as everybody understands what you're talking about.
The thing on the left that's going off the screen is a pineapple siphonophore. So are we on transect? We seem to be stalled again.
Yeah, we're going on the move. We're just sorting out a few things.
Gotcha.
Yeah, we're about 50 meters from where we marked the start.
Wow. Cool.
So did you, Tom, did you see the Lizzie Duncan's chat to us about where we're heading?
Because she says we're heading for a boring looking canyon.
Yeah.
So, sorry, Ben.
That's why they gave it to us to do the boring canyon, and she got to do the fun stuff.
I'm adding it to our report.
See, and that's a big fin eel pout. You can tell by the
30.
slope of the
Yeah,
head.
okay. We're not crazy. No.
There's another eel pout.
Yep. All those guys are big fins.
Big fin for a little fish.
Yep. That's actually, they do have huge pectoral fins for the size of the fish. That's where they get their name from.
Yep. Well, we'll probably make good time over this terrain.
That's the positive outlook.
Yeah. Silver lining to everything in life.
It's interesting. We've been over mud many times today, and we still haven't really seen any sea pens.
Yeah, if you guys want to go slightly upslope at some point as we make our way to the next target, it's doable with us.
Yeah, what we're doing now is fine.
Okay, great.
There's another.
So see the rounded head on this guy?
Yeah.
And he has like
Is that
the
slender?
kind of stripes across him. No, that's a Rex.
Oh, it's
He has
Rex
the
sole.
stripes that don't go all the way across. And there you can see the long pectoral fin that gives you the idea that that's a Rex. Or
Okay.
that's the definitive character.
So a rounded head, stripes that don't go all the way through, and a long pectoral fin.
Yep,
Let's see
and
if
the
I can
long
remember.
fin is the one that's definitely the key, because no other flatfish here has that.
Rex being a king, right?
Shall I
You know,
tell the
I don't
story
know
about
why it
how?
was called Rex.
Yeah.
Sure,
It
you go
doesn't look
tell
very
the story you
kingly to
want.
me.
Well, the story of how they get their eyes on one side of their head. They start
Oh.
out as larvae with eyes on both sides of their head, and when they settle, so as larvae they're up in the water column, and when they settle to the sea floor, depending on the species, the eye that is on the light skin side that's sort of on the substrate, it moves across the head to the other side.
Aren't there
Migrates.
left-eyed and right-eyed, like the
Yes,
pleuronectiformes and then the Bothidae or something? One of them starts on the
exactly.
yeah.
And I'm going to let Tom, I think Bothids are right. Maybe I 50% right.
You're 50% right,
Yeah.
but you're also 50% wrong.
Okay, so did I flip the coin in the right direction? Or I can look it up really quickly now.
Well,
The eye is half full.
you tell me
And
what you think the answer is.
Well, I'm looking it up.
I think
They're
pleuronectiformes
left-eyed.
is left-eyed.
So I was wrong. Bothans are left-eyed. But I had learned when I was in college that there's also species where rather than eye migrating over the front side of the head, so like across the nose, if you will, the bridge of the nose, that there's also species where they sort of travel right through the body.
I have not seen that, but you know, there's so much diversity that I can't say that for sure, that there's not a species that does that.
Yeah.
But it seems like it would lose its vision at the point that it was inside its body for a while.
That's true. That's true. But what's interesting also about flatfish is you can see the counter shading so well that the part of the body that is on the seafloor is white and the top side is colored, so that when they're swimming through the water, as a predator looks up, they would disappear against the white of the light that's shining down.
And there's also no reason for it to have any color on the part that's facing the mud. So on the left, we have a spot prawn, the one with the 2 dots. There's a commercial fishery for them, and they're actually very tasty.
What do they market them as? Do you know the name?
Spot prawn.
Oh, okay. Easy.
So a lot of the people in our area just sell them off their boats. So you go to the boat and get spot prawns.
And it's a pot fishery, correct? They
Yes. Hook and line fin is really tough.
Yes, but they don't use nets.
Yep. Nope. So that's why
it's
in
like
the Monterey area, it's tough, because when you see buoys, you don't know if it's just a crab pot or if it's a string of spot prawn traps. So when you're running vehicles underwater, especially tethered ones like the ROV, you've got to be careful for things like that, because you don't want to get it caught.
Yeah.
Hey, we're coming up to a Christmas tree that I don't even see if it's alive. It doesn't even look alive, but it has a black coral crab sitting on it.
Yeah, Christmas tree of Christmas past.
Yes. There's an octopus on the left.
Oh, yeah.
There's a Sebastomus rockfish. That's a group of about 10 species, and it's tough to tell which species it is, but it looked like it might have been a simulator.
You say Sebastes?
It is a Sebastes, but it's in the Sebastomus subgenus.
Okay.
So these are all split nose, and if you look in the middle of the fish, it has like an upside down U,
Mm-hmm.
and that's one of the easy characteristics from far away. That's a bank rockfish sitting next to the rocks, just kind of down and right of the lasers.
With the white band.
Oh, the one that's hiding
whited
a
out
little
one.
bit.
Yep.
Yeah.
A lot of diversity.
So we're
with the
seeing-
rockfish.
Yeah, we're seeing some change in the rockfish species now, so maybe we'll start to see some other corals. Or some corals. Looks like there's a little Asbesta-pluma, one of the white stick sponges we collected down below.
Is that the carnivorous sponge?
Yes.
Cool.
Basically looks like Velcro if you put it under a microscope or zoom in. It's got these little hooks on it, and
To
it
be
catches
carnivorous.
crustaceans. Yeah, you have to be small to worry about it, but once it catches them, it just like absorbs them.
Sufficient.
Yeah, I guess it is. That's not the way I want to go, but you know.
With a sponge, it's sufficient.
Yeah, getting eaten by a sponge.
It's pretty lame.
If you can't outswim a sponge, that's not good.
Right? They don't even have body symmetry or anything.
Yeah. So the coralish passing on the right is a Christmas tree coral with zoanthids growing on it. So zoanthids are our colonial coral, similar to like in a colonial anemone. Look at that, fox and rockfish.
Earning their name.
And to your eyes, there's a diversity of species that we're seeing, or in that one shot?
Of which, the rockfish?
Yeah.
I see two species really I see. Up in the upper left, there's a hake, the silvery fish with the kind of mud halo around him right now. But most of these rockfish on the bottom are split nose. When we're off transect, if somebody wants to zoom in or if we have the opportunity, their upper lip is split. So there's a divot between the left and the right side, and that's why they get the name split nose.
Is that for like chemosensory functions or smelly?
I don't have any clue of why it's that way.
A wafting appendage?
Yeah. And so it's when they used to be like a rhino, or no, it has two horns out in front. That's not a rhino. Rhino has one horn, that would work.
A two-horned rhino.
Something with, there you go, yeah. That's why they're extinct. The two-horn rhinos, they had those. But the nice thing about the black coral crabs is you can see them in the black coral even when you can't tell the coral's there because they're just up in the water column with that crab. Go ahead, Lisa.
No. It wasn't important. So you recognize the black coral based on that crab that is sort of sitting on top of it, even though most of the distinguishing characteristics are gone?
Yes, pretty much. And it's the only one that's going to have that kind of skeleton left over. And then we have these three California king crabs right here, or spiny crabs. The yellow guys.
Definitely can see the spines.
Yep. I've noticed that the common names for a lot of species are pretty easy when they figure them out. We'll call it the spiny crab. And the black coral, because it has a black base, black skeleton. So, see, like if you look at this crab on the right, it's in the tree, but you can't really see the Christmas tree as well as you can see the crab up in the water column.
Yeah.
What it looks like.
So it's a dead giveaway.
Yeah, there's not too many swimming crabs except blue crabs, and that's not a blue crab.
No, I mean, dead. The dead gave away that it's on a that's a Christmas tree coral.
Yeah, Christmas tree corals are oftentimes I don't want to say hard to see, but the polyps and the structure of them make them more translucent until you get up close. So when they're off in the distance, you can't necessarily see them as well, unless they're a huge one. And then we have this swiftiest species in front of us by the crab, almost dead center. It's a little red fan that we're just going over. And there off in the distance, you see the crab on the tree. And all these, as far as I can tell, look like split-nose rockfish.
Even the very white one that we're almost going over, it's just a.
Yeah, so you see the size of the eyes. Proportionally, they have bigger eyes.
Yeah, they're
So
very
you can
big.
see, yeah, so that's one of the other characteristics of split nose. So that's what I would say that, though some species, I mean, every fish is slightly different, so it's hard to say 100%, but you can get a good high probability if that's what they are.
115, 75 left.
Was that 115 left or 115 gone?
We've come 115 meters, yeah.
Okay, thank you.
Yeah. We should be able to keep pretty straight till the end of that. All those sea urchins are gone now.
Yep.
What we were saying? Yeah. The white spines.
They're fragile urchins.
Say that again?
They're called fragile urchins.
Oh, okay. Are they actually fragile?
They actually are. When you trawl them up, they just come up in pieces.
Oh.
So their species name is fragilis. And here we get a petrale sole right here with this big dark spot on him. And the two little white dots.
That spot's diagnostic.
It is pretty much. With that pattern, it's pretty much the only one you see like that.
Oh.
There's quite an active fishery for petrale sole, and there's a petrale sole spawning ground up to the north off of Santa Lucia Bank.
Where we would have went and sampled, but we lost a day because of the weather.
That's right. But who knows? The weather gods might be good to us, and
Yep. You never know when you're out here.
that's right. You never know. Yes. Yeah.
Yes, you did.
Ah, stink. Yep, that was quite an unexpected find in 2018 when we looked at some of the AUV imagery and found these fish all stacked up on top of each other. Definitely not how we're used to seeing them.
And that's a Dover sole. It's making all the mud in the water. Stirring it up, trying to get away. But it's interesting when you look at the fish that things that can get away don't even seem to care that this big, giant 2-ton vehicle is coming past it, and they don't care. How much does Hercules weigh?
Around 5 tons.
Oh.
I believe that's correct, right?
So that's even worse.
Definitely bigger than a bread box.
Maybe
Yeah,
I'm
well.
thinking Argus, but pretty sure it's Herc.
That's pretty heavy.
It's heavy, yeah.
And here,
It is crazy to think
here
about,
come,
though. Like these organisms live their entire lives in silence and darkness, and then suddenly a loud, super bright monster thing appears out of nowhere.
and they don't run away. And now we're back to the urchins again.
Yeah.
But you know, when you watch sort of these alien movies with humans, some of them depict humans as sort of, you know, dumbfounded and starstruck as they look up at the alien starship that descends on Earth. So maybe that's how they sort of view this. So, you know, out of the norm that it doesn't faze them.
So we should look for fish rock drawings on some of these rocks.
Yes. Well, they are rockfish.
Ah, true. And that's a Dover sole we're going over with the 2 dots. There's also been some little poachers that we've seen, or I've seen, but they're all been about 10 centimeters or so, so they're harder to spot till they're just about off the screen. And that's a big fin eel pout, no pattern at all on the side of his body. If you could look at his front of his dorsal fin, there's a black dot.
Big fin eel pout.
What do the eel pouts feed on, Tom? They got a funny shaped mouth.
They basically eat things off the bottom, so little crustaceans. I don't think they eat too many worms. Most of the Dover soles, the deep sea soles, a lot of the soles will eat the worms and the in fauna, but most of the fish that are sitting on there are eating small crustaceans, and then larger fish will eat the smaller fish and things like that.
So they'd just be scavenging if we weren't, or are they kind of ambushing things?
The eel pouch, you mean? They're
Yeah.
probably just going along, either waiting for something or just when something comes along, they get it. Most of the deep sea things, you know, they're opportunistic. Some of them are actually specialists, but most of them will eat almost anything.
Anything that comes their way.
Generally, I mean, the flatfish are like Dovers and deep seas pretty much just eat worms and some other in fauna, but mostly worms. You can't see all that right now, but if we actually dug up the bottom, you can see a lot of critters that are living just under the surface, allowing up all this nice organic material in this mud. Guy's got some long claws on him.
Yeah, so we have about, still 30 meters to go. But then there will be a slight bend, Jess, so we can continue north until we see them.
Roger. Roger that. And what's the distance to the next target, 8K?
Yeah, still going to be like 700, 600 meters.
Roger.
600.
So once we finish the transect, then if we're still in this type of bottom, we can just kind of pick up the speed and head toward there. And I don't think we need to slow down unless we run across some hard substrate where we want to look.
Roger that.
Yeah. We do have a slope map for you, but I'm not sure we have backscatter for this site.
Yeah, it's going to be a hit or miss.
Okay.
Is that a Rexol?
Yes.
That is.
Yes.
I'm actually looking at the fish in the lower right was a chili pepper rockfish.
Oh,
But he's gone now, but I had to wait till he got
a
close
red
to
hot
ID him.
chili pepper.
If you say
Caliente.
so.
I used to live 2 doors down from the lead singer.
Really?
Yeah, back in the day when they were just starting out.
That is like the most California thing ever.
I
I was just
know.
thinking that.
Well, yeah, when you live in LA, what can I say?
Yeah, no, I love it. It's just perfect in its stereotypicalness.
But you weren't able to sleep well at that apartment.
It was a house actually.
Oh.
Back when you could rent houses for it wasn't crazy expensive yet.
Where, in Beverly Hills? Where were you?
It wasn't the worst part of Los Angeles, but it was not Beverly Hills for sure. Not that they would live there either. Maybe now, but back then.
Was this LA proper, or was it one of the surrounding cities?
No, it was LA.
Look, we have
Downtown was
Paragorgia.
not far.
There's some Paragorgia made it on transect now at the lasers. There's one of the pineapple siphonophores.
I was just looking up that siphonophore, and the only paper I could find about those benthic siphonophores near California said that they come up a lot during, they collected a lot of samples during a sewage monitoring project. Yep. And the species we could be seeing is
10 meters.
Dromalia alexandri, which has a very large pneumatophore, which is that kind of floaty top part of the pineapple or the hot air balloon.
They're still, are they still Vicynectidae? The like Vicynec siphonophores? With the swimming bells and the
You want to
singing
add the species name to the chat?
Yeah, I'll put it in as a maybe.
Yeah, great. That would
Modality is
help.
the family. And apparently there are about 15 known species.
But when Abby, when you told us the species, the genus and species name, I totally understood why Tom calls it pineapple siphonophores.
Yep.
I got it.
We're going to continue on this heading. We can do another chip move in, and then we can finish the transect.
Yep. Yeah, so we're done with the transect now. We're at 200 meters.
Roger that.
We can continue on this going north, or we could just head directly to AK and go up this slope. Sure.
I think that depends on what science wants. Do you guys want us to continue on this contour as we make our way over?
Just,
let me look, see where AK is.
Yeah, it should be up on the upper right in high pack.
Yeah, we can just go up slope. We don't have to necessarily go, we'll go straight up.
Roger that.
Get to some different habitat maybe.
Yeah.
Roger.
Is that good for you?
Yep.
We only have like, what, hour and 20 minutes left, so.
Yeah, let's jet.
Yeah, I'll go a little upslope from Argus.
Yeah, I'll wait for you. It's going to be 050.
050, okay.
And it's going to be flat for about 100 meters, and then it will be a slope.
Okay. Let's do in 30-meter steps.
Yep.
Actually, I'm pretty good here if we're going to go 050.
Okay. Yeah, the ship is super responsive, so I don't mind waiting.
Yeah.
Something with it looks like very large pectoral fins, or it's a flatfish moving just to the right of the lasers. I can't see it now.
Yeah, I don't see what you're talking about yet, but I'll keep looking.
No, it disappeared in the rocks.
Yeah, that's all right.
That's a magician fish. I see where the mud was kicked up.
Yeah.
So we have a short spine thorny head to the left, and with its spines up.
Huh.
So when we were deeper, we were only seeing really long spines.
Yeah, that's
where
what I
the
was going to
third-
say.
Yeah, the third spine in the first dorsal fin is longer on a long spine, and it's basically the same length on a short spine. It's the only definitive way to tell them apart.
Do they co-occur, or are the long spines really deeper and the short spines are shallower? Or that doesn't matter?
Yes, generally. And the short spines also segregate by size. So the shallow ones, the little ones are shallow, and the biggest ones can be deep. So if you're down deep and you see a really big fish, that's most probably a short spine, whereas all the other ones will be long spines.
Okay.
It's all very practical ID information for us.
Yes.
Yeah.
And when you look at this skate,
Oh yeah,
this skate with this long nose makes it easy to tell what kind that is.
You don't say.
you can tell
It's
by
a
the
long
gas.
nose skate?
Marlene, do you
It is a long nose
Yeah.
skate. Sure.
Sweet.
Nose to the nose.
He really looks like he's trying to camouflage into the sand here.
And just like sharks, he has claspers, so he's a male.
All right, Marley, come wide, please.
And so just because I'm a proud grandpa, I'm going to say, hi, Emmy and Theo, because my grandkids are listening right now, so I get to shout out to them.
That is so cool. And we get to
Yep.
say, your grandpa is quite special.
Yeah, your
Do you
grandpa
know
is pretty
how
cool.
many fish species? Yeah, that how he can just, recognize fish just when they're 90% or almost their entire body is covered in mud, and he can recognize them.
Remember what you said at the beginning. Just say it and people will believe you too, so that, you know. I'm just saying it, even though it's true.
No, I do know that you have scruples, so.
A little. There's another one of the dromalias, or the pineapples.
Oh yeah.
Siphonophore. Yes, I'm able to see things a little more because the sun's setting, so the room's getting darker. It helps. Whereas you guys in the van, you're like pretty much in dark all the time, right? Just the monitor light, basically.
That's correct,
It's
yeah.
by design.
Yeah. Well, that's right. Whenever I've been at sea doing this, that's what you're sitting in the dark. Makes it way easier to see things on the perimeter.
Then you come out, it's like getting out of the cave,
Mm-hmm.
Yes.
blinded.
If you switch over to channel 3 on Nautilus Live, you'll see,
there we can see it.
inside the van.
Ooh,
Pretty
look,
much
there it
just
is.
all monitors.
I don't have that many monitors.
How many monitors do you have up? We should share with each other our control stations at home, our home stations.
I have 2 laptops and one of them has a monitor attached. So I basically have 3 screens going right now.
Good. I have 2 laptops and each have an external monitor. So I have
That's
4.
better than me.
Yeah, 4 light. Well, yeah, doesn't seem to make up in my ability to ID, but I'll try.
So my granddaughter, she's asking a question on why is the ground green?
Okay,
So
very
basically,
good question.
That is. Basically, there's little organisms that live in the water column, and when they die, they sink to the bottom. So there is what it's called organic matter all over the bottom. And that creates, instead of looking like brown mud, it creates green mud. But then we're also looking at artificial light. And it depends on how the light and the cameras work together to change the colors of things that we see on the bottom.
Green soda.
That's my quick answer.
Marley, want to do a
It's a
partial?
Sprite.
Is that can? A can of?
Sprite.
Can of
It's
Sprite?
Sprite.
Yeah.
I just want to see the...
Where is it?
It's below
A green can of Sprite. Oh, this is the shell hash. Should we try and collect this?
We should do a
Is it
scoop
the
here.
shell?
Yes, that would be good.
You want to stop?
This is shell hash, I
Nav,
believe. So I think we can do it as a scoop or as a sediment core.
Since it's not really consolidated, a scoop would be better for this.
Okay,
Yeah.
great.
So Lizzie wants to find a dense patch of hash, or the densest one we can find there.
Would you prefer...
Maybe some to the right?
Some to the right? Roger. Does it have to have this bacterial matting, or would you prefer not to?
No, she just wants this. That's, I mean, this area looks a little more dense than the other area.
Roger. Maybe center screen on the lasers now?
Yeah, that looks good.
Great.
And if we could zoom in on the shells before we pick them up, or the area.
Definitely.
Right, and so Lizzie's also asking if those are brachiopods, but I'm not seeing.
Well, they're all dead,
These
so
might
if the
be-
shells are
Yeah, these might
split
be
in half.
remnants of brachiopods.
Marley, you want to push in there, center screen?
How many scoop samples can we take in a dive? Lindsay would like to know.
Gonna come a little wide so we can get the extent of the.
Sure. Is that good?
Thanks. That's great. Let's bounce it a little bit.
And they look like clams, not brachiopods.
Remember that. We have one scoop available, and I don't believe it's been used, so we don't have 2 scoops.
No, it's not Raisin Bran.
No 2 scoops of Raisin Bran. Is this Zoom all right for you guys?
At least
Should we
somebody
start
got
setting
that.
up?
Yeah, this is great. Thanks.
All right, Marley, go ahead and come wide. And you only want the surface layer, or would you want, how deep do you guys want to go?
So Lizzie, you're in the chat, so tell us how deep you want to go.
From reading the instructions, I don't think he indicated the contact from the Santa Barbara Museum of Natural History.
Yeah,
I'm
so
look in
just
the protocol.
she's saying medium.
Yeah, because I don't think he indicated. I'm going to look real quick at the protocols.
Yeah, and Lizzie said he didn't indicate. So yeah, just get whatever like a medium scoop would be. There you go.
Medium scoop of ice cream, Raj.
That's right. Not Raisin Bran though.
Yeah, upgrade.
Yeah, we've already had our breakfast for the day. Okay, would you mind switching over the port cam there, please?
Okay, so this one, just to let all the women on here know, my granddaughter keeps asking if the woman's voice on the picture is grandma. So you've all been grandmothered into my family, I guess.
Oh, that's sweet.
We'll take that as a big compliment.
Yeah. So I'm reading here, it just says avoid pure mud. That is the only instruction on the shell hash.
That's not pure mud.
Yep.
Yeah. I was assuming. Roger.
Oh, it's very kind.
I see a scoop.
Thanks, Kim.
It looks quite large. I was not expecting. It's like a bucket. I really thought like a scoop of cereal. I don't know why I thought that.
We have that sometimes
So
too.
I was really thinking that you were coming out with this, like, teaspoon scoop.
So for those of you watching, if you go to the quad view or channel 3, you can see the scoop that we're talking about. And now it's on the main camera.
I want to stay down there, does it? Oh, yeah, we got our pick of the grab here. Before it went on its own little journey there. All right.
So would this have been a graveyard of? So I studied coral reef biology, and so if you saw something like this, you would know that you were in an area where there was an octopus, and this is the remains of a octopus
Marley, you want to push in there,
meals.
please?
So what causes something like this to have this
Come a little
field
wide.
of shells?
That's great there. All right, so
My guess.
oh, sorry guys, one moment.
Yeah.
Sorry.
So no, it's all good. So I'll grab basically the top layer of the shell hash, and there isn't really a disclosed depth, but does that sound reasonable to you?
Yes, it does.
Roger. All right. Sorry for the interruption. Please continue.
Oh,
You know
no,
what? You
guys
take
were
precedence
first.
while you're doing stuff. So,
Yeah.
but Lisa, I would say that this could have been a seep, and these could be shells that were left over from the seep, because I don't see many of these shells on the rocks. So something else
Right.
had to have been here to keep these alive. And so that would be my guess. But I know exactly what you're talking about because you see the shells even diving in Monterey around octopus dens. So
thank you for that explanation, because I was just wondering, like, why would they all be concentrated in one place? So that makes sense.
it also seems like a little wash right here, if I remember coming up to it. But I don't know if that has anything to do with it. But if you see washes, things will congregate at the lowest point when they roll down a hill. So.
And do you want me to grab another scoop and shake this out, or they actually want most of the sediment in it?
I think.
Go ahead, Lisa.
From reading the request, you can shake out the mud. But
shake out the mud.
Yeah, shake out the mud.
Roger that. Car wash time. Gotcha.
Shake, shake, shake.
Are you want to come a little wide there? We don't get
We're
a
gonna
whole...
break into song now. The 4 to 8 watch is the fun watch. That's all we know.
Yeah.
4 to 8 is great.
So we got one scoop in there. If we shake out the mud, we can go for 2, or do they happy with 1?
I'm happy with 1.
Roger that. like it's ready for the liftoff. All right. Call that well shook.
Well, you've stirred it up so we can't say anything, so that definitely is a good shake.
Which one do you want?
Thanks, Kim.
So if you're watching at home, you can watch on the quad view or channel 3. They're putting the scoop away.
In BioBox F?
BioBox F, indeed.
No pressure, Jess. It's just thousands of people watching you do this.
Yeah.
Roger that. Hello, everyone. All right. She might not close. I can try giving it a little tap here. Sorry, Kim. There we go.
So the scoop typically resides in BioBox F?
F or E, depending on if we feel like we want vowels or not.
So the larger ones. Okay, great.
Yeah, generally the larger ones. Cool, she closes. Thanks, Kim.
Got it.
You can do anything. You got that coral, everything's downhill after that.
Yeah.
Scoops are fun. All right. Back on our way?
Yep. I'm going to continue on 055.
055. Roger that.
+5.